Hello everyone, welcome to today's live broadcast, Formulation Selection and In Vitro Screening for Oral Bioavailability Enhancement. This is Adeline Siu, Science Editor of Pharmaceutical Technology, and I'll be your moderator for today's event. We are pleased to bring you this educational webcast presented by Pharmaceutical Technology and sponsored by Capsugel, now a Lonza company. Capsugel, now a Lonza company, designs, develops, and manufactures a wide range of innovative dosage forms for the biopharmaceutical and consumer health and nutrition industries. Capsugel's unique combination of science, engineering, formulation, and capsule expertise enables customers to optimize the bioavailability, targeted delivery, and overall performance of their biopharmaceutical and consumer health and nutrition products. The company partners with more than 4,000 customers in over 100 countries to create novel, high-quality, and customized solutions that align with the customer's evolving needs and benefit patients and consumers. For more information, please visit www.capsugel.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. Please also note the resources available from our sponsor located in the green resource widget on the dock at the bottom of your screen. If you have any technical problems viewing or hearing this presentation, please click on the question mark help widget in the dock at the bottom of your presentation window. Concluding today's webcast, we invite you to participate in a brief survey from our sponsor. The survey will automatically appear on your screen. I would now like to introduce today's speaker. We are pleased to be joined today by Dr. Michael Grass. Michael Grass is a senior research chemist at Capsugel, now a Lonza company, where he develops methods, conducts testing, and analyzes data for a wide range of pharmaceutical formulations. His expertise includes formulation development of challenging compounds, dissolution testing, and physical stability of amorphous dispersion. Dr. Grass, thank you for joining us today. Please begin your presentation.
Thank you for the introduction, Adeline. So, as Adeline said, today I'm going to be talking about formulation selection and a little bit about in vitro screening for oral bioavailability enhancing technologies, and really focused on the approach that we take here at Capsugel for defining technology selection as well as formulation development. I'll start a little bit about discussing the problem statement at hand about doing compound-driven formulation development. And then I'll mention, at least briefly, a few different common technologies that we take in terms of formulating these compounds. And then I will touch on in vitro testing and different in vitro tests that we can use to formulate the compounds that we have. And then I'll discuss a few case studies that we've done here at Capsugel Bend, and if there's time, we'll talk about all three. So as many of you are aware of, more than 80% of compounds now within the development pipeline are either BCS Class 2 or Class 4 compounds, meaning that their solubility is lower than that would be required to dissolve in the intestinal fluid. And that means that there's a lot of room for development of formulations that increase solubility. There's another classification system called the Developability Classification System, or the DCS, and that divides the Class 2 compounds into Class 2A and Class 2B. Class 2A being dissolution rate limited and Class 2B being solubility limited. So there's several properties of compounds that we need to look at when we're deciding on a technology to use and a formulation to develop. And those include, in gray, things like physicochemical properties like solubility, hydrophobicity, molecular weight, and such. Also in blue are some of the pre-systemic metabolism and permeability effects that we need to think about when formulating a compound. And then also in orange on the left, things like what does the dose need to be to reach efficacious levels, and what are the pharmacokinetics of the drug and pharmacodynamics that lead to the PK profile that we need for that compound. So when we take those molecular properties, we need to combine those molecular properties with a product concept, typically given to us by a client. Things like, is it going to be a solid or a liquid dosage form? Is the product going to be taken once a day or twice a day? How many units are okay to be taken by the patient, and the size of the tablet or capsule that's being taken? So a lot of those product concepts, along with those molecular properties that I discussed and some predictions of properties and performance, go together to define a problem statement for a compound. And that problem statement I've shown there as the BCS classification, but it goes well beyond that in terms of what are the potential risks of developing this formulation. Things like chemical stability of the molecule, physical stability of the molecule, and its particular performance attributes, both in vivo and in terms of manufacturing. We take that problem statement assessment, and then we use that to define what technologies we will look at to develop, and what kinds of formulation approaches we will take, and also will help us define what types of in vitro testing we'll do in addition to in silico and in vivo testing. And then that really makes for us a loop that we call product development. So a few of the common bioavailability enhancing technology platforms are listed here. In blue on the left-hand side, I've listed some of the solid state alterations, so changing the form of the compound or the particle size, things like polymorphs, solid dispersions, making the compound amorphous, as well as size reduction into microcrystals or nanocrystals. On the right is kind of a different type of approach where we solvate or complex the molecule using things like co-solvents or lipid-based formulations or cyclodextrins to make complexes. And in the middle is kind of an approach where we've made kind of a new crystal entity, either a co-crystal or a salt, which can also improve bioavailability. So I'll mention a few of these in the next few slides, how we select between them, and how we think about the way that we formulate each of these technologies. So when we think about different problem statements for bioavailability enhancement, we often think about things such as dissolution rate, solubility limitations, and permeability limitations, as well as metabolic limitations to the correct PK profile that we're looking for. When it comes to dissolution rate and solubility, there are several technologies which may, depending on the compound's other properties, fill these problem statements. So dissolution rate limited, obviously size reduction is a great way to increase dissolution rate. Also the formation of a salt, if that's appropriate, or a co-crystal, as well as amorphous and lipid formulations, can all address dissolution rate limitations. And many of them can also address solubility limitations. When we get to permeability or metabolic limitations, the options are a bit lower, and when one option that's not listed on here, because it's considered more applied to drug product rather than formulation, is the formation of a prodrug, which is often a good approach with permeability limited compounds, where a prodrug may be more permeable. So this plot here is one example of a plot we might look at when choosing a technology for a particular compound. On the x-axis is log P, or the lipophilicity of the compound, here ranging from 0 to 10 along the x-axis. And then on the y-axis is the aqueous solubility. This is given in the aqueous solubility, assuming a 100 milligram active dose, and then that can be shifted up or down depending on the actual dose of the compound. But you can see for the highly soluble compounds, crystalline forms are typically well absorbed without the need for bioavailability enhancement. And then we can go to nanocrystals as we become dissolution rate limited. Amorphous forms work very well for very low solubility compounds. And lipids have particular advantages for high log P compounds. So we see it there in the bottom right. There are, of course, a lot of other factors that influence what technology we might select, and we'll discuss that more in the coming slides. So one option here, the first option I talked about is making salts, polymorphs, and co-crystals. This is really applicable for class 2 and class 4 compounds, BCS or DCS class 2 and 4. Some of the advantages of these forms, they're highly precedented. We have a very high dose potential. There's typically fewer excipients required. And the manufacturing is quite straightforward. Some of the challenges is that without some of the more enhancing excipients, precipitation can be a problem. And sometimes these compounds convert even in the solid state to a lower energy form on stability, which can decrease bioavailability of these forms. Moving on now to another one is particle size reduction, typically of the most stable crystalline form, though this can be done with higher energy crystal forms as well. This is particularly applicable to DCS class 2A, which is dissolution rate limited. Though if it's combined with another crystal form, obviously that could apply to the full range of class 2 compounds. There are a lot of approaches to size reduction, as shown in the upper right-hand corner. Things from ball milling to microfluidics and precipitation approaches. And all of these have different places depending on the properties of the molecule and what it's amenable to. The biggest advantage, of course, of making nanocrystals is the dissolution rate enhancement that one achieves by making very small crystals. It also has a very high dose potential because potentially fewer excipients are required. And this can be a particularly good approach if an aqueous suspension is desired. Some of the challenges that we face with making nanocrystals is sometimes the lengthy and high cost of production, as well as stability on shelf life and the fact that we don't get a lot of solubility enhancement. So this is truly an application towards dissolution rate enhancement. Moving on to some amorphous forms, there are a couple major ways of achieving amorphous dispersions, and the first one I'll mention is hot-melt-extrusion. Hot-melt-extrusion is a great way of achieving an amorphous dispersion with good solubility and dissolution rate enhancement, which we get for all amorphous dispersions. HME has high throughput, well precedented, and a small manufacturing footprint. Some of the challenges with HME are the narrower space in both compound and excipient properties. Because of the high heat used in HME typically, we have to make sure that the compounds are thermally stable and have a melting point or a dissolution point into the excipients that we use that's appropriate. Another way of making amorphous dispersions is by spray drying. Of course, here in Bend is one of our core technologies that we use is spray drying. The applicability to compounds and to excipients is typically a bit broader than it is for HME. Otherwise, we see a lot of the same advantages that we see for HME for an amorphous dispersion, though we typically end up with smaller particle size and therefore faster dissolution rate. On the flip side of that, it is a solvent-based process, and that can be a limitation for manufacturing and manufacturing costs. The final formulation I'm going to mention here today is our lipid-based formulations. Lipid-based formulations are typically used, again, for class 2 and class 4 compounds, though sometimes for class 3, depending on the type of permeability limitation and the properties of the compound. Some of the real main advantages of lipid-based formulations are their effect on the biology in the intestinal tract. So in particular, their ability to induce a fed state and the increased bile concentration that comes along with that, which can be a very large advantage particularly for high log P compounds. Another advantage is the ability to increase lymphatic uptake of a compound. Again, particularly for compounds with log P greater than 5. That lymphatic uptake can bypass first-pass metabolism through the liver and reduce metabolism of some of the highly metabolized compounds without having to worry about whether a patient has actually had a meal or not, as it provides the lipid itself. Lipid formulations also increase solubility and dissolution rate, although they typically do not increase drug activity as high as an amorphous dispersion, which can be a positive or a negative depending on the application. The biggest challenge with lipid formulations, of course, is the limited compound space. The compound typically needs to be soluble in the lipid formulation, and we typically end up with either a liquid or a semi-solid formulation, which may be either a physical or chemical stability risk depending on the compound. So in this table here, I tried to just summarize a few of the advantages of lipids versus solid dispersions and vice versa, and really where we can identify lipids being critically different from amorphous dispersions. As I've indicated here, the critical lipid differentiators are really in these biological factors, things like inducing the fed state and lymphatic absorption, which can be very critical for certain compounds. For amorphous dispersions, on the other hand, they tend to create and sustain a higher drug activity and higher drug solubility, as well as being possible to combine with modified release formulations more easily, and tend to have higher dose per volume than a lipid formulation. And so those are some of the key advantages of solid dispersions, and they can guide us to selecting a technology depending on the compound and the problem statement. All right, so now I'm going to mention a little bit just on a few in vitro tests for bioavailability enhancing formulations. This is not meant to be exhaustive, but just give a couple examples of tests that we run here for different problem statements. So when we're thinking about in vitro dissolution testing and in vitro testing in general, one of the big questions that we have is how is our drug product going, the properties of our drug product going to impact the plasma concentration that we observe in the clinic. And of course, there's a lot of complex physicochemical processes, including absorption, disintegration of a drug product, as well as dissolution in the stomach and in the intestine, issues of pH, solubilization, and then permeability and metabolism. And so what we really need to do is consider all of these physiological properties, things like what are the fluid properties, what is the pKa and the presence of micelles, biosalt micelles in the fluid, what are the volumes and hydrodynamics of those fluids, as well as what does the permeability look like. And we need to combine that with many of the drug substance properties, its solubility, its pKa, its lipophilicity, along with many other drug substance properties, and then the properties of our actual drug product. What is the particle size distribution? How do my excipients behave in the stomach and in the intestine? And how does my drug product disintegrate and dissolve to release into the intestine? And so based on that, we can design several different tests at several different stages of development to get at some of the key properties of the compound, the drug substance, as well as the drug product. So I'm going to mention just a few tests that we use in formulating here at Capsugel. So one of the tests that we use prior to developing a formulation is to understand a drug's propensity for increasing solubility in the amorphous state, as well as its ability to be, for that increased solubility to be sustained by a polymer. And of course, this is really targeted towards identifying possible amorphous dispersion formulations that could be helpful in formulating a compound. So in this test, what we do is we dissolve a drug, typically in an organic solvent, or possibly in a high or low pH aqueous solution, and we inject that into an aqueous buffer of interest. And so the plot at the bottom, the pink curve shows our total added drug concentration, which we add over time. The x-axis is time, and the y-axis is either concentration or scattering. And what we observe, the black trace shows the concentration of drug, and we see that it increases linearly as we add drug until we reach a critical concentration where the solubility plateaus or the concentration plateaus. And this is the concentration at which drug is no longer going into solution but is going into a second phase. So the red curve shows the scattering that's occurring, and at that same point we see an increase in scattering where the red curve ticks up off the baseline. And that corresponds to the picture you can see in the upper right where we have some scattering off of particles, and these are amorphous drug particles, and they indicate the concentration of the amorphous solubility. And we can compare that to the crystalline solubility to understand our maximum enhancement that we can get by making an amorphous dispersion. And then the second thing we notice, and we can run this test in the presence of many different polymers or other excipients, is we can observe how long that supersaturated drug concentration is sustained. And so the middle section where that black curve is flat is termed sustainment. That's where we see the drug sustains before it begins to crystallize to a lower energy form. So that's one critical test for us prior to starting formulation. And then once we've formulated, we have several different options for tests. Of course, many of our standard dissolution tests, non-sink and sink dissolution tests, which I won't mention here, and I'll instead mention a few other more specific ones that we use in particular cases. So one of the tests that we like to use is what we call a membrane test, or sometimes referred to as a flux test, where the drug formulation is put into one side of a vessel, which is separated by a membrane from what we call a receiver compartment. So there's a donor compartment and a receiver compartment separated by a membrane. And typically when we run this test, that membrane is a lipid-filled artificial membrane, not unlike a Pampa membrane. And we look at the drug concentration over time in the receiver compartment to compare formulations. And this is particularly useful in cases where it is difficult to determine the true concentration of dissolved drug from a formulation because of scattering or because of a lot of highly dispersed but not dissolved drug. This can also be useful in cases, and I'll talk a little bit more in detail about this later, when the permeability of a compound is limited by aqueous boundary layer diffusion, in which case truly the diffusion of drug into the receiver compartment, it can be a combination of not only dissolved drug but also drug that's bound in micelles, as well as small colloids or liposomes or other small complexes which might contribute to diffusion across the aqueous boundary layer. And then this can be a very good way of not only rank ordering but also understanding relative differences between formulations. Another critical test for lipid formulations is what we call the digestion test. And in the digestion test, the idea is that we actually digest the lipid formulation that is used to carry the drug. So a lipid formulation, one of its key benefits and also challenges is that the excipients are digested in the intestine by enzymes. In particular, in this test, we use pancreatin to digest the lipid formulation. And depending on the composition of that lipid formulation, as well as how the drug interacts with it, can change the rate of digestion and therefore the supersaturation of the drug and sometimes precipitation of the drug. So for some formulations where the drug can be quite supersaturated and stable as an emulsion in water, as soon as it becomes digested, the drug becomes more supersaturated as the solubility tends to decrease in the digested lipids, compared to the original lipids, and we can get drug precipitation. And so this is a very critical test for comparing lipid formulations. And the last test I'll mention is one often referred to in the literature as a artificial stomach duodenum test, or goes by other names as well. And the idea here is that we start with our drug formulation in a stomach or gastric compartment, and we transfer it into a fixed volume that we call the duodenum. So in this picture, there are some vertical syringes, which are our artificial stomachs. And then there's a little cell that material is transferred into, which is our artificial duodenum, that we keep at a constant volume while we continually flush new gastric fluid or FASF into that chamber. And what we can use this test for is to really see the intricacies of a compound which tends to crystallize on transfer from the stomach to the duodenum. So the data shown here on the right is for a weak-base compound, or lotinib, and in black is the drug dosed alone. So because it's a weak base, it dissolves in the stomach and transfers perfectly along the blue line, which is the theoretical transfer rate, for about 15 minutes, and then undergoes very rapid precipitation. However, if we dose this drug simply with some HPMC physically mixed with the drug, it follows the red curve, where supersaturation continues for about an extra 5 or 10 minutes, and then when it precipitates, it's a much more gentle precipitation, and the solubility remains much higher throughout the test. And so we can use this test to really identify formulations which can inhibit precipitation. So we like to combine all of these tests and many more and use them in a way that we look at as a way of doing a sort of formulation, risk-based formulation approach. And so here you can see in the box on the left, we do some initial preliminary assessment, which is based on many of the basic properties of the compound, and we use those properties to identify particular problem statements. So this is just a sort of a theoretical example for a compound which maybe is a weak base, so therefore has gastric pH-dependent dissolution highlighted as a problem statement. Maybe its solvent solubility is an issue for certain formulations. And we can take all of those problem statements and define what technology we're going to use. In this particular case, it was a spray-dried dispersion. And we can make a few formulations which we then screen through a set of some standard tests, as well as tests identified from those problem statements. So we're using that problem statement risk assessment to define our testing strategy. And then through that screening, we can redefine our problem statements shown up in the upper right-hand corner, where we've identified certain problem statements which were initially identified, which don't appear to be true challenges for this compound, but we've also identified a few extra problem statements that weren't noticed in the initial preliminary assessment. And we can use that in kind of an iterative approach to really come up with an ideal formulation prior to the more costly efforts of scale-up and clinical trials. So I'm going to shift now to talk about a couple case studies. The first case study I'm going to briefly talk about is increasing the bioavailability of a low-solubility NSAID, celecoxib, which goes under the trade name Celebrex. So celecoxib is a sort of a moderate melting point, relatively low molecular weight, and moderate to high log P compound, log P4. And you can see in the bottom left, I've shown a new plot of selecting between SDDs and lipids. And again, this doesn't tell the whole story, but we can see here with log P on the x-axis again, and in this case, melting point on the y-axis. Melting point being a critical parameter for lipids, as with higher melting points, we tend to get lower lipid solubility. We can see that this compound falls kind of right in between them, though very much on the edge of where we might consider a lipid formulation, and a little bit more in the SDD box. And so in this particular case, we formulated a spray-dried dispersion. So there's some literature data out there on formulating a lipid dispersion as well that I'll mention. So this compound has high permeability, and even in the crystalline form, it's fairly well absorbed. So here is a summary of some of the in vitro dissolution data of an optimized solid dosage form at 50% loading. So actually pretty high loading for a solid dosage, or for a spray-dried dispersion. On the left is the in vitro dissolution data. In red is the bulk crystalline API, and in the green shows the dissolution performance of the spray-dried dispersion, both initially and after 6 months of aggressive stability conditions at 4075. And what we can see here is that indeed the SDD gives very enhanced dissolution performance in terms of extent of dissolution, and sustains that high concentration for at least 40 minutes through the test. On the right-hand side is the clinical PK data comparing the SDD dosed either as a suspension in purple or as a tablet in green to the commercial dosage form, which is a crystalline powder and capsule. We can see an almost 3-fold enhancement in Cmax and about a 2-fold enhancement in AUC in the clinic. So here an optimized SDD performs quite well and gets close to full bioavailability in the clinic. We did not formulate a lipid for this particular compound, but there is some data in the literature on optimizing a lipid formulation and comparing it to, again, Celebrex, the commercial formulation. And you can see here there's a fairly modest increase in Cmax observed for the lipid formulation with a bit faster onset, so showing that the increased dissolution rate is making an effect, but something is limiting the lipid formulation, either the extent of dissolution or sustainment, which is keeping it from seeing the same enhancement that we see for the amorphous form. And then here's another interesting formulation which we looked at after making the SDD. We looked into also forming another amorphous formulation of this compound, being an amorphous nanoparticle. And in this particular case, it's an amorphous nanoparticle of the active with an insoluble polymer ethyl cellulose. And this provides an interesting platform by which you can actually modify the activity of drug that's achieved by changing the ratio of celecoxib to ethyl cellulose, which changes the solubility that's reached in the intestinal media while keeping almost instantaneous dissolution rate. And you can see here the effect of that is that the onset, or Tmax, is much shorter for the red curve, the nanoparticle formulation, than it is for the SDD shown in green. But overall AUC doesn't change too much. All right, so the second case study I'm going to show is another one where we look at both amorphous dispersions and lipids for a low solubility compound with a large food effect. This compound has greater than a 10x food effect, so the bioavailability in the fed state is about 10 times that in the fasted state. The dose of this compound is about 400 milligrams, with fairly low fasted state bioavailability, but well absorbed in the fed state. And the problem statement here was to develop a formulation that would decrease the food effect and could be dosed without respect to food. And you can see here on the map on the left again we are in that intermediate ground between lipid and SDDs, now a little bit more perhaps the lipid side in terms of this plot. And another indication for this compound is something that we often use in SDDs to look at the ability of a drug to crystallize, is the Tm over Tg shown there in pink. And the Tm over Tg is the ratio of the melting point to the glass transition temperature. And in this case, the value of 1.4 is quite high and suggests that this compound has a large propensity to crystallize. So we might think that it's a precipitation risk. So in the development of an SDD, we looked at several formulations, and while there's some differentiation in terms of how much total drug gets dissolved from different formulations, in this case in a test where we transfer from gastric environment on the left-hand side of the graph at early times to an intestinal environment on the right-hand side, we see that all the formulations perform quite well compared to the crystalline active in a fasted state test. But they all precipitate relatively quickly. Prior to 30 minutes, they're all precipitating. And so we see that unlike the last example, where our sustainment was at least greater than 40 or 45 minutes, here it's closer to maybe 10 to 20 minutes. And in fact, as we developed our dissolution tests to be a little bit more robust in terms of finding how much dissolved drug there truly was, we found that the precipitation was even more extreme than that initial test suggested. So we were able to develop a UV-based test that could distinguish between dissolved drug and finely dispersed nanocrystal and precipitate of the drug. So when this drug precipitates, it precipitates to a very fine nanocrystalline form, which can be hard to separate from the dissolved drug by standard methods. And we can see here that, in fact, the dissolved drug shown in orange spikes up at about 2 to 5 minutes and then is almost at its baseline level but prior to 10 minutes. So this drug, in fact, while supersaturating quite well from an amorphous form, is very difficult to sustain. So we moved on and formulated this drug also as a lipid. Looking at several different formulations and using the digestion test I described earlier as the final way of selecting our optimum formulation from a list of formulations that were developed. And so in this test, the drug is initially dispersed in something similar to a fed state medium, and then at the dotted line, where you can see the vertical dotted line is the addition of pancreatin enzyme, and the formulation begins to be digested. And we can see how drug sustainment follows versus time. And you can see here the orange and the gray formulations sustain that drug to at near 100% of the dose for at least an hour, while the two other formulations shown in pink and blue, the drug is precipitating over time and crystallizing to its crystalline form. So one of these key formulations was selected along with the optimized solid dosage form, and were tested in a canine study in a fasted state. And so there are 5 box plots here shown for a canine study, here showing just AUC, total AUC for the dog study. On the far left is the commercial crystalline product in the fasted state with very low bioavailability. The second one is the SDD, and we see that while we get some improvement relative to the crystalline form, the improvement is not as high as we were hoping in reference to the fed state commercial product shown on the fourth one. So quite modest relative to what we can achieve closer to full bioavailability in the fed state. The lipid formulation, the one in the middle labeled T6 in the fasted state, achieves high bioavailability, although as you can see there's quite a bit of variability in between individuals in the data. And then the last one on the right, which I didn't discuss, is a solid lipid formulation that was developed, a semi-solid lipid formulation of the drug, which gave performance and AUC similar to that of the liquid lipid formulation, and could be a good option here. So in the end, we were able to reduce the food effect from greater than 10 times to less than 2 times food effect, and increase bioavailability in a fasted state, more so with a lipid than with a solid dispersion in this particular case. So the last case study that we have, we'll try and go through fairly quickly, is focused more on developing an in vivo predictive dissolution test for the antifungal Itraconazole. So in this case, I'll focus just on a spray-dried dispersion formulation, and how we selected an in vitro test to try and be in vivo predictive. So in this particular case, as you can see here, we're kind of on the edge of both an SDD and a lipid due to its high log P as well as relatively high melting point. So the first test that we ran, as I mentioned, is the amorphous solubility test. And we can see that although the crystalline solubility is exceedingly low in FASIF, so that was on the previous slide, you can see there is less than 0.1 microgram per milliliter, the bottom number on that table. In the amorphous form, we get an enhancement of about 100x. It's nearly 10 microgram per milliliter in the amorphous form. So we do expect to see a very large effects by formulating in the amorphous state. And in fact, the commercial product for Itraconazole is called Sporanox, and it's an amorphous spray-layered dispersion of Itraconazole with HPMC that gives reasonably good bioavailability. But we wanted to see if we could improve on Sporanox with a different amorphous formulation, and how we might test that in vitro. So in a basic in vitro dissolution test of a solid dispersion of Itraconazole, we see that in a typical assay using microcentrifuge separation of the solids from the dissolved drug, we see a very high concentration, and in fact a concentration much higher than what we know the dissolved drug concentration can be from the amorphous solubility. So the graph on the right shows that the actual dissolved drug and micelle-bound drug is a small fraction of the total drug measured, and the majority of drug measured in this test is in the form of drug polymer colloids, shown with the blue bar. And so we wanted to develop a test that would indicate whether these drug polymer colloids had a large impact on the bioperformance of formulations of Itraconazole. So the next slide just shows a little bit more information about these colloids that are formed. They have a size of about 150 nanometers, which we determined by both DLS and nanoparticle tracking analysis. And you can also view them, in fact, by microscopy, simply by drawing out a suspension of them on an SEM post and visually looking at them through the SEM. So the test that we developed to compare formulations, amorphous formulations of Itraconazole, was a membrane-based test where the formulation was dissolved into a donor compartment in various intestinal buffers, and the concentration in the receiver phase was monitored. Through a set of tests that are described in the paper highlighted here, we determined that Itraconazole is aqueous boundary layer limited, and therefore the presence of colloidal drug and micelle-bound drug can potentially have a big impact on the rate of permeability, both in vitro and in vivo. So this is a picture of the test as we have set it up. We run this in parallel. We can run eight vessels at a time, and the drug is formulation and donor media is placed into a vessel, and a receiver vessel with the membrane is placed on top inside that outer reservoir, and the drug concentration is monitored in the receiver phase using a UV probe. And in the upper right, you can see a zoomed-in version where we have a lipid-filled membrane separating two aqueous phases, the donor and the receiver compartment. So for this particular test, the in vivo study was going to be in rats, and so we wanted to at least somewhat match both the fluid composition and the rate-limiting step to absorption in the in vivo and in vitro case, even if we don't can't match identically things like surface area to volume ratios. So for rats which have fairly high bile, we chose 27 millimolar sodium taurocholate with lecithin. So this represents about 2% CIF in aqueous media, and a pH well above the pKa of itraconazole, which is in conjunction with the rat intestine. And so in both cases, in vivo and in vitro, we expect dissolution rate to be very fast compared to absorption, and we expect that the rate-limiting step will be the aqueous boundary layer diffusion. So we compared 4 formulations in this test, and so the black formulation shown at the bottom here is the commercial formulation Sporanox, and then we looked at 3 different formulations with different grades of each HP and CAS, which resulted in different concentrations of colloids, as well as different levels of sustainment. On the left, you can see the dissolution curves, so monitoring concentration in the feed using HPLC over time. And on the right, you can see the concentration of drug in the receiver phase over time monitored by UV-Vis. And you can see that we can, while there's a almost 100-fold or even more difference in the concentration observed in the dissolution assay on the left between the highest concentration reached in a formulation and Sporanox, the actual receiver phase flux is only about 2.5 times that of Sporanox for the optimized formulation. So in this test, we can see that the effect of these colloids is very much muted compared to the total concentration of them. And this is because they are much larger than truly dissolved drug and only contribute fractionally to permeability across the aqueous boundary layer. This slide is just showing that we took several formulations and several different media, and were able to, in fact, have a very good correlation between the calculated flux based on the concentration of dissolved drug, micelle-bound drug, and colloidal drug, and the measured flux in vitro. So we have a good correlation, and we're able to understand the driving force for increases in flux based on formulations that are developed for this compound in vitro. And so the idea was to go and look and see how this plays out in vivo. And in a rat study dosed at 50 mg per kg, we see that in fact the difference in AUC between the different formulations is measurable, but not as extreme as what we see in vitro. But if we plot on the right-hand side, the y-axis being the maximum flux in vivo, so that being the maximum absorption rate observed in vivo, versus the flux observed in vitro on the x-axis, all relative to Sporanox, we see a pretty good 1-to-1 correlation between the in vitro flux and the in vivo measured flux in the rat, which indicates that we have a good in vivo predictive test for which we can optimize formulations based on. So in summary, formulation selection, there are a lot of options out there, and it can be challenging to select the correct technology and within that technology the correct formulation. But we have many in silico tools and in vitro tools, and if we take the time to really think about and understand the true problem statements and the true challenges for absorption of a particular compound, we can design the right tests that help us develop an optimized formulation with the right technology. Continuing to develop these in vitro and in silico models will continue to help us in both technology selection and formulation development, as well as building up some historical databases where we've compared formulations. I showed 2 examples where we compared formulations, but certainly as we look at more and more compounds with complementary technologies, we'll be able to understand further how to select them. So thank you very much for your time, and I will hand it back off to Adeline for a poll question and wrap up.
Right. Thank you, Dr. Grass, for this interesting presentation. So before the Q&A session, I would like to ask our audience to participate in a brief polling question. Please click directly on your screen to submit your answer. Here is the question. Would you like capsule gel to follow up with you after this webcast? So please click the answer on the screen. The question is, would you like capsule gel to follow up with you after this webcast? Thank you for participating in our first poll. I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. We'll try to answer as many questions as we can. So here is our first question. Dr. Grass, you briefly mentioned the incorporation of amorphous dispersion into modified release formulations. Can you describe typical modified release approaches for amorphous dispersion?
Yes, thank you for the question. We have developed modified release formulations for several different projects for amorphous dispersions. One of our key technologies for that is what we call SCT, or a swellable core tablet, which involves making a bilayer tablet where there's an active layer, typically with the amorphous dispersion mixed with a swelling excipient such as polyethylene oxide, and then a polymer layer, which has a higher molecular weight, PEO, that swells but does not dissolve. And that swelling layer, as water comes into the tablet, pushes active formulation out of the active layer through a hole that's been drilled in the active side of the tablet with a laser. And this is a commercially available technology that we have developed for modified release, in particular controlled release of amorphous dispersions. It can be a good way to sustain high supersaturation for long amounts of time in the intestinal tract.
Thank you. So let's move on to the next question. Can you comment on screening techniques for biologics, especially peptide drugs?
Yeah, that's a great question. My experience is a little bit less with biologics and peptides. We've worked on it a little bit. Oftentimes, as I'm sure you're well aware, these peptide drugs are sort of fall into the BCS classes 3 and 4 areas, and it's particularly challenging, especially when these peptide drugs are quite water soluble and have very poor permeability. Many of these technologies are challenging to develop, permeability enhancement in a reliable and reproducible way. And so the peptides can be especially challenging for oral delivery.
Hey, thank you. Next question. So one of the audience has observed that a couple of times that they experience similar concentration in donor results in different acceptor flux. The question is, do you rely on flux from time 0 to 6 hours or over a much longer time frame?
Yeah, that's a good question. So I think the question was similar concentrations in the donor resulting in different acceptor flux. Yes, I think that can be due to many different things, and it depends on how the concentration in the donor phase is determined, what separation method is being used, whether it be centrifugation, ultracentrifugation, filtration, and what size filter is being used, and then the properties of the drug and the formulation. Those can all impact what that concentration in the donor phase really means. And then also there's the interaction of the drug with excipients, which can sometimes limit the availability of that drug to permeate across the membrane. And so the second part of the question, do we rely on flux from time 0 to 6 over a much longer time frame? Typically, yeah, we are looking at just the first few hours of flux. We have seen some interesting cases where the flux over longer periods of time tells us maybe a much different story or something interesting, but always then also begs the question of whether the membrane is still intact over a much longer period of time than 6 hours. So, but yes, the short answer is we typically look over the first few hours.
Thank you. So the next question is, how do you work with clients to define the problem statement for a compound and identify formulation approaches?
Yeah, this is definitely a very collaborative effort that we use, where we work with the client and you know their needs for a product, which go beyond just the compound properties, but also deal with things such as, you know, what does the presentation of the drug need to be? Is the patient population typically on PPI or, you know, proton pump inhibitors, which can modify the pH of the stomach? Is our population typically elderly or sick in other ways and on other drugs which may have drug interactions? So it's really important to fully understand the full scope of the problem statement when formulating, which often we bring the technical side of you know, understanding compound properties and developing that into a formulation, but the client typically has a much better holistic view on all the other problem statements that might be involved.
Thank you. The next question, you mentioned the formation of colloids in both lipid formulations and amorphous dispersions. So the question is, what is the difference in the colloids between these technologies?
Yeah, so the question was about the passage of colloids through the membrane. And this slide here, slide 40, that's up on the screen right now, has a reference to a paper that was published where we really tried to look into what is the effect of colloids and micelles and the impacts that they have on permeation through the membrane. And what we were able to determine through a lot of testing of different formulations and different compounds is that the colloids do not pass through the membrane, but they can help diffusing across the aqueous boundary layer. And in a follow-up publication to this one, we also explored, for the specific case of itraconazole, the impact of colloids quite in depth. And we were able to show that while they don't pass across the membrane, they can increase boundary layer diffusion, particularly when the micelle concentration is low. So particularly like in the fasted state, or in cases where micelle concentrations are very low. And in particular in cases where solubility is exceedingly low. So if you remember, even the amorphous solubility of itraconazole is about 10 microgram per milliliter, and it is necessary that the solubility is quite low for these colloids to have a large impact on the flux across the membrane.
Thank you. Also, Dr. Grass, in one of the examples, you mentioned that you used UV method to determine the absorbance or concentration and avoided interference of nanocrystals and precipitation in the solution. So, could you explain the method a bit more?
Yeah, so that's a method where the nanocrystalline drug particularly tends to have a different absorbance than the dissolved drug. So a little care has to be taken in developing a method where you can separate those two signals. But it is possible to determine sort of the characteristic UV signal of the nanocrystalline drug and the dissolved drug, and then to deconvolute the signal. And so we did that work quite some time ago using a pion fiber optic system and doing all of our own analysis and deconvolution of the data, though I am aware that Pion's new software, I think that they're coming out with soon, they have said that it will be capable of doing such deconvolutions in the software package itself. And so that's kind of the method that was taken there.
Right, thank you, Dr. Grass. Unfortunately, we are out of time. I want to thank the audience for attending and participating in today's event. I would also like to thank our sponsor, Capsugel, now a Lonza company, for making today's webcast possible. Today's program will be available for on-demand viewing through September of 2018. You will receive an email from Pharmaceutical Technology alerting you when this webcast will be available for replay. We invite you to forward the announcement to any colleagues who might have missed today's live event. As a friendly reminder, concluding today's webcast, we invite you to participate in a brief survey from our sponsor. The survey will automatically appear on your screen. We'll see you next time. Thank you. Bye.